Hi, everyone. Welcome. I'm actually going to introduce Dr. Fahrner. She's going to go first. Dr. Fahrner is the director of the Epigenetics and Chromatin Clinic at Johns Hopkins. She's an assistant professor of genetics and pediatrics at Hopkins, and she is my wonderful colleague. She has a great interest in the Mendelian disorders of the epigenetic machinery, of which KAT6A is one, and glad she's here.
Thank you, Dr. Harris, and thanks to the KAT6A Foundation for inviting me to speak today. I just wanted to point out, I'm really glad that we could all meet in person to be at the third international conference for KAT6A and KAT6B.
Just need to go.
Okay, that's okay, thank you.
And the middle one is come right.
All right.
If you want the microphone, take it off.
Oh. All right, sorry about that technical difficulty. So today I'm going to be giving an overview of KAT6A syndrome, and I'll be sharing some clinical, genetic, and epigenetic perspectives. So the objectives for today are to introduce concepts of genetics and epigenetics, to understand KAT6A syndrome as a genetic disease with epigenetic consequences, to provide an update on current knowledge of the molecular changes and clinical features in KAT6A syndrome, and to introduce our multidisciplinary Epigenetics and Chromatin Clinic at Johns Hopkins. So just to kind of start from the basics and make sure we're all on the same page, our bodies are made up of billions of cells. Each cell contains genetic material in the form of DNA. The DNA contains about 22,000 genes, of which KAT6A is one. Genes determine traits, so things like eye color, hair color, how our brain develops, these are all determined by genes. And each gene is a set of instructions or code to make a protein with a specific function, or one or more functions. And the DNA sequence or code is made up of 4 bases, A, T, G, and C. And genetics refers to this DNA code. So genetic variants or mutations are mistakes in the DNA code that cause disease. The genes encode proteins that then have particular functions in the body. And so the KAT6A gene encodes the KAT6A protein, which then goes on and has a function, which I'll talk about in a little bit. So variants or mutations in the KAT6A gene cause KAT6A syndrome by affecting the ability of the protein to function properly, and that has implications for many different systems in the body. So as many of you probably know, KAT6A is an autosomal dominant disorder. This means that only one copy of the gene is affected and the other functions normally. So you may recall that we all have 2 copies of each gene in our bodies. And so in KAT6A syndrome, we have 1 copy that's not functioning properly and the other copy is functioning properly. So there is some functional protein that is made. So I thought it could be helpful to go through some of the different types of variants that we see in KAT6A syndrome, because I think people talk about these a bit, and I'm not sure how much families understand what they mean. And so I'm showing here just the normal setting, where you have the KAT6A gene, which has no mutations, the KAT6A protein is functioning properly, and there's no KAT6A syndrome. Sometimes you can have a missense change in the DNA, so it's like a misspelling. You can think of it like a misspelling of a word. And if you have a missense change, this can lead to reduced function of the KAT6A protein, and this leads to KAT6A syndrome. But very few individuals actually have these missense changes. More individuals actually have truncating, what we call truncating changes. And these can be early truncating or late truncating. And in early truncating variants, we think that essentially no protein is produced. And in late truncating variants, there may be an abnormal protein that is produced, and it could affect the normal copy or sort of poison the normal copy. And so the later the truncating variant occurs, if it occurs in the last 2 exons, this seems to correlate with more severe phenotype or features of the disorder. So this is a schematic of the KAT6A protein showing the different variants that have been reported in affected individuals. So the black type shows protein truncating variants, and those that occur in the last 2 exons, above and below the green and orange parts, are the later truncating, and others are the earlier truncating. And then the red type shows the missense variants, which you can see are far fewer than truncating variants in this condition. So thus far, we have talked about the fact that DNA is our genetic material and contains our genes. The genes make proteins in cells with particular functions. And variants or mutations are mistakes in the DNA code that alter it. And mutations in genes like KAT6A cause particular disorders, in this case, KAT6A syndrome. So what does KAT6A do? I said that proteins have various functions in cells. So what does it actually do? So KAT6A switches on genes in cells. So I'm just going to kind of show you how that works. So this is a schematic of the DNA, and our DNA is made up of genes. But the DNA does not exist alone in cells. It's actually wrapped around histones, and together the DNA and histones are called chromatin. And so this chromatin can be marked by something called epigenetic marks or chromatin marks. And the particular mark of interest in KAT6A syndrome is acetylation, and that's indicated here by the green hexagon. So KAT6A switches on genes by adding this chemical group called acetylation. And here I'm just showing that KAT6A places this mark. There are other components in this system that remove the mark as well. And what this mark really does, this acetylation mark, is when it's present, we tend to see the chromatin is more open and the genes are turned on or switched on. And if it is absent, then the chromatin is more closed and the genes are turned off. So KAT6A mutations may cause the syndrome by disrupting, in part, by disrupting the acetylation mark and gene expression. So you can imagine if you have a KAT6A protein that's not functioning properly, that this could affect levels and placement of this mark and could lead to differences and changes in gene expression. So, but we think that this probably happens at many genes throughout the genome. And so this slide just kind of shows a properly functioning KAT6A writer of this acetylation mark. And just as a schematic, we think that it may place acetylation on genes 1, 2, and 3, but it may not affect genes 4 through 8. But if it's not functioning properly, then it can't place this mark on genes 1, 2, and 3, and it can't switch these genes on. And so if these genes are involved in growth, learning, or craniofacial development, these are the things that are going to be impacted. And this will happen at many genes throughout the genome. So in essence, we think of KAT6A as a genetic disorder because it results from a mutation or a variant in a gene, but it has epigenetic consequences. We think it probably disrupts epigenetic marks and gene expression at many genes throughout the genome, and that may be what accounts for all the different features that we see. One interesting and potential thing that could happen in the future is, there are drugs that can treat, that can reverse epigenetic marks. And so because we have a loss of this acetylation mark in KAT6A syndrome, there are epigenetic drugs that could potentially restore that mark. And so this is something that may be something in the future that could help. So in summary of this second part of my talk, so KAT6A is a writer of epigenetic marks, particularly acetylation. Mutations in this gene may cause particular genes to not be switched on correctly. KAT6A target genes may determine the clinical features of the syndrome, and in the future, it may be possible to tweak epigenetic marks for some sort of therapy. So now I just wanted to go through the features, the clinical features of KAT6A syndrome that are caused by all of these molecular changes that we've talked about. So as many of you, as you all know, individuals with KAT6A syndrome have many neurobehavioral findings, and I'm not going to spend much time on those because Dr. Harris will be talking about those in detail. But they have developmental delay and intellectual disability with speech delay that is present in essentially 100% of affected individuals. They have low muscle tone, behavior problems, and in rare cases, seizures. They also have eye findings, so individuals with KAT6A syndrome can have strabismus, or kind of where one of the eyes will sort of deviate outward or inward. They can have ptosis, where the eyelid is partly closed, vision problems. Some individuals will have craniosynostosis, which is premature fusion of the skull, which can lead to differences in the head shape. They can have GI findings, so there's often GI dysmotility, so things don't move through the gut as they should. And this can lead to feeding difficulties, reflux, constipation. Also, about half of the individuals with KAT6A syndrome have congenital heart defects, and these are typically defects in the septum, so atrial septal defects or ventricular septal defects, but there are others as well. And individuals can have frequent infections, and this can be caused by immune cell problems or hematopoietic cell problems. And then sleep disturbance. This is another thing that these individuals have. And then growth abnormalities, including a small head size and potentially being small at birth. There's also some common facial features in KAT6A syndrome. You may recognize some individuals here from this paper. So the most common features that we see as, that other people may not notice, but as geneticists, we notice these things. They can have a broad nasal tip, a thin kind of tented upper lip where it sort of looks like pulled up almost, narrowing of the temples here, a prominent nasal bridge. They can have a short and flat philtrum, which is the space between the nose and the mouth. They can have epicanthal folds, which are some folds inside the eyes, and they can also have low set and posteriorly rotated ears. So this paper by Kennedy et al. Has shown that the prevalence of some features can differ based on the location of the KAT6A truncating mutation, as we talked about. There could be early or late truncating variants. And what they found, and this is sort of a busy slide, but what you can see is that the dark gray bars indicate the presence of a particular feature, and the light gray indicate absence of the feature. And and you can see that there's early versus late. So you can see that in the case of microcephaly, neonatal hypotonia, a thin upper lip, feeding difficulties, reflux, constipation, congenital heart defects, and frequent infection, all of those things occur more frequently if there's a later truncating mutation. Similarly, the developmental delay and intellectual disability may be more severe with late truncating mutations. You can see that there, if you look at the side, that's the intellectual disability score. And if you look at the lower score, so that means there's milder intellectual disability, that tends to occur with early truncating mutations, but the higher, more severe intellectual disability tends to occur with the later truncating mutations. And similar findings were found with just looking at developmental delay. So how do we manage KAT6A syndrome in the clinic? It's important that they become involved in early intervention and get appropriate educational supports. And these may include sign language or communication aids. Echocardiogram is important because about half of individuals with KAT6A syndrome will have a congenital heart defect, so it's important to take a look. Regular eye exams, because they can have many eye findings, which we discussed, and they can develop over time. It's also important to symptomatically treat GI manifestations. Also important to consider an immunology workup because some individuals will have immune dysfunction and it may not be immediately obvious. Also considering sleep study if there are problems with sleep or signs of obstructive sleep apnea, and melatonin can be used as a treatment. Abdominal ultrasound is also important. I didn't mention this because it's not as frequent, but there can be structural anomalies of the abdominal organs, so it's good to have one abdominal ultrasound at some point to make sure there's nothing there that needs to be addressed. And then brain MRI may be considered in some cases. And audiology evaluation is very important because of the speech delay. It's important to know if there's any contributing hearing loss to that. And then I'll just end by talking about our Epigenetics and Chromatin Clinic at Johns Hopkins. So it was founded in 2012 by Hans Bjornson. And then I joined shortly thereafter, and it became multidisciplinary when Dr. Harris joined our clinic. And now I'm directing the clinic. And anyone who comes to our clinic will see myself as the clinical geneticist, Dr. Harris as a neurologist and neurodevelopmental specialist, and Jackie Britton as our genetic counselor. And this is just a wheel that shows these Mendelian disorders of the epigenetic machinery, of which KAT6A is one, and I've highlighted KAT6A there, because these are the disorders that we focus on in our clinic. And we have seen hundreds of individuals with these disorders, and we follow a fair number. I would say KAT6A is sort of one of our top 5 diagnoses that we see in our clinic. So we have seen quite a few individuals. And we would be happy to see anyone with KAT6A or KAT6B and be happy to help in any way that we can. And I just want to say thank you. And this is a picture from our conference in 2018. So glad to be together again with everyone. So thank you so much, and I'd be happy to take any questions.